Welcome to everybody for attending our first Research and Health webinar for 2026. I know we normally do the first Wednesday of the month, but we thought this for January it would be a little bit too soon. People would still be in Christmas mode. So I hope everyone had a good break and a happy new year to everybody. I'm very pleased to welcome Dr. Kristin Seltmann here to be with us for this first webinar. So Dr. Seltmann is a senior scientist at ETH Zurich, and her work has focused on skin cells and keratins, keratinocytes, particularly relevant for people with EB simplex, where it's a genetic change in the keratin that is causing the problem. So she's going to talk today about strengthening and signaling proteins in skin cells. Just for a little bit of housekeeping, as it's a webinar, our attendee cameras and microphones will be switched off. The webinar will be recorded, and we don't want to accidentally record anyone. So please don't make private recordings or take screenshots. We'll have the Q&A working for questions. Submit them as we go along, and we'll have them asked and answered at the end. The chat will be enabled if you want to make comments, and I think that's probably it. So I have to apologise that we are planning a webinar for next month, but I can't confirm the speaker just yet. So welcome again, Dr. Seltmann. Very pleased to have you with us. Would you like to share your presentation? And I will disappear off the screens. Thank you, everybody.
So welcome, everybody. I'm very pleased and honored to have this opportunity to share my research. And what I will do today is basically share my research over the years. And I'm very happy to see Thomas in the crowd, because I will actually also talk about my PhD work first, and then I will continue with my PhD work, which I did in Germany, University of Leipzig. And then I moved to Zurich, where I'm still located today, and I will talk about my skin research, which I'm doing here. So first of all, I think you're mostly all aware of that, but I would like to remind you why we actually have skin as such an important tissue. Sorry for that. Okay. For why skin is actually so important. So overall, we all know that the skin is a very important barrier for mechanical damage, against viruses, bacteria, but also irradiation. So it's actually crucial to keep the skin barrier intact, to keep the protection of the skin against of the human body. This is a classical outside-in barrier function, but we also have an inside-out function, which is actually tremendous if we lose the skin, or a large amount of the skin first, which is to retain water inside of the body, but also keep the body temperature intact. This is also a very crucial function in the skin. And last but not least, it's also a sensory organ for touch, or if the feeling gets too much, then it's of course causing pain. So we need also to sense that, and in addition to this, we can also sense other environmental factors like the temperature. I will come to that a little bit later. Now, how is the structure of the skin composed of? So we have all these three parts. So the epidermal part, the dermal part containing the fibroblasts and immune cells secreting a lot of extracellular matrix. And underneath we have the adipose tissue and blood vessels, as well as later on also muscle cells. I will concentrate today on the basal layer of the epidermis, which is located here. And this basal layer connects the epidermis to the dermis, where it's separated by the so-called basement membrane. So therefore, those cells in the basal layer, they have two very important functions. First of all, they need to connect to each other to keep the barrier intact, but also they need to provide quite a tremendous strength underneath to adhere actually to the basement membrane to keep the epidermis attached to the dermis, which is indeed a problem in diseases like EB for patients. One important reminder of the epidermis is it's a stratified tissue. We have a keratinocyte in the basal layer, which contains a stem cell compartment. Eventually, they differentiate and move on into the upper layers, the spinous layers, stratum corneallosum, and then also in the stratum corneum. Doing so, they're actually changing their properties of the proteins quite tremendously up to the stratum corneum, which is mainly the cells mainly build up lipids to build up this barrier against the outside to the outside environment, and also lose their nuclei and eventually are completely shedded off. This process is called skin homeostasis, and is very crucial to keep the skin always intact or over time. So what is special or most important structurally on this keratinocytes of the basal layer is kind of depicted here. So to keep a barrier of the epidermis intact, we need basically two major structures. We on one hand need this attachment to the basement membrane. For that, we have focal adhesions connecting the extracellular matrix to the actin cytoskeleton, or we also have the hemidesmosomes, which connect the extracellular matrix to the keratin cytoskeleton. And then for the cell-cell contacts, which are extremely crucial for keratinocytes, we have the adherence junctions, the desmosomes, and here not depicted also the gap junctions. And one factor which is also very important for the skin is that it needs to be resistant against mechanical stress. And for that, we have actually three cytoskeleton components which keep the cells in resistant and able also to react to different mechanical stresses. We have in all cell types, we have the actin cytoskeleton and the tubulin cytoskeleton, whereas very specific for cell type, we have the expression of the third cytoskeleton, the intermediate filaments. And keratinocytes, they are called keratinocytes because they contain keratins, and actually to a huge amount, they express the keratins, and these keratins form the keratin filaments. So the keratin cytoskeleton is a very tight network in keratinocytes, and they are in particular interesting because this is a heterogeneous network. We have always heterodimers of one type 1 and one type 2 keratin in the cells forming this network. And what is interesting on them is depending on the differentiation status, depending on the tissue, and depending even on the state of the cell, meaning if it's under stress or if it's in a disease state, they actually express different keratins. And we also know that point mutations of keratin genes can cause epidermolysis bullosa simplex. And as most of you probably aware, this is a disease where the epidermal is kind of getting fragile, and every single mechanical damage leads to the skin blistering, which then fill out of liquid, and it's a very it's a chronic inflammation which then is occurring in those patients. And I was starting my PhD work exactly from this, from the question, what is actually the function, the overall function of the keratin cytoskeleton in keratinocytes? Why is this not an easy question to answer? This is mainly because of this second bullet point here, which is that keratins can express keratinocytes can express several keratins at the same time. Meaning if we, in the lab, deleting one keratin, what will happen is that by a redundancy, another keratin is most likely taking up the function. So we are having in the end not a keratin-free cell, we actually have a cell with altered expression of keratins. So to kind of overcome this, the lab of Thomas Magin deleted the entire keratin type 2 cluster genetically. I will not go into experimental details, but I will show you later what the summary of my work and was. And I focused in this keratin-free cells at that time at cell matrix adhesion. And I'm not going in too much details, but just that you have a broad overview. Hemidesmosomes are the cell matrix adhesions which are directly linked to the keratin cytoskeleton. And I like this electron microscopy image here very much because you can nicely see how the keratins form this filamentous structure and keratinocytes and are then connected to the hemidesmosomes, which you can see here, which appear as plug structures. So they're really dense structures, a dense protein complex. And why is this? This is basically because the extracellular matrix, which is depicted here in the scheme, is linked via integrins and intracellular interacting proteins like plankton and BB230 and all the others. These interacting proteins here are actually large proteins. So plankton, for example, is 500 kilodalton in size. So all these kind of proteins make this generate this complex. And we were interested if we delete the keratin cytoskeleton overall, what is happening actually to the cell matrix adhesion, migration, and invasion, and also other properties. Exactly. And really very briefly, the experimental model where a mouse keratinocytes which were either wild type keratinocytes having a normal expression of the keratin cytoskeleton, here this is a K5 staining, or non-expression at all. Or then what we also needed to figure out is to this relatively severe deletion of the entire family, of the entire type 2 cluster family, can we actually rescue the phenotype of the keratin-free cells by bringing back K5, K14 in those cells and see if we have a rescue, meaning if we can actually have similar results like we can see in the wild type. And the results we obtained were the following. So first of all, what we saw is that if keratinocytes lose keratin, the cells migrate much faster in comparison to wild type cells, which you can see here. And this is a collective migration. Collective migration means that you have a complete confluent cell layer, then you scratch it, and then you follow how this two cell clusters, if you like, move together, which is kind of similar to a wounding, even though we miss something what we have in vivo, like for example immune cells and so on. But we try to sample this classic migration of keratinocytes, which is relatively more collective than single, since keratinocytes are in this contact to each other, which is very important for them as well. Now, then we also did single cell migration experiments to see if something is altered in the directionality. And there we also saw that, first of all, they're much faster, but also that they move in very different directions. So they are not so much interested on the neighboring cells, which is also an unlikely behavior for keratinocytes to do. So that was quite interesting. Now, regarding the cell matrix contacts, we focused on hemidesmosomes, since these are the structures which connect the keratins to the extracellular matrix. And hemidesmosomes are assembled like it's depicted here. So you usually have plankton and beta-4 integrin co-localizing to build up this very plugged, large structures of the cell. And this is the case in those cells. But if you now use this mutant, this is actually an EBS mutant, what we first of all see is that we don't see a proper keratin cytoskeleton in those cells anymore. We actually get this vesicle structure, so the cells are not able to form this network. And this has consequences for the hemidesmosomes as well. You can see that plankton is completely differently localized compared to the control. This is also true for beta-4 integrin. And in the end, this causes that there is no colocalization possible between plankton and beta-4 integrin, and therefore no proper hemidesmosomes can be formed, which also explains why the keratinocytes in those patients are unable to have a proper adhesion to the basement membrane. Then, and one other very important factor of keratin loss is invasion. We know that in skin cancer, one of the first hallmarks is the decrease of the expression of keratins, and eventually even alter the expression from keratin, which is classical epithelia expression, to vimentin, which is a classical mesenchymal expression of cells. This is a process so-called EMT. And so therefore we was wondering, could there be a change in the invasion properties of those cells? And what we saw in an invasion assay was we saw more invasion, but with similar proliferation of these cells. Just quickly, let me explain how this is done. You have a transwell assay, which has a porous membrane, which is depicted here in green. And then you seed the cells on top, and then they eventually squeeze. They really have to squeeze through this pore, so they have to get a little bit smaller and move further down. And then we quantify how many cells have been able to do that. What is important is also this squeezing process when it goes through, because what you can see here down here is that the keratinocytes, the keratin-free cells, the knockout cells, look actually a bit ruptured. We had also measured biomechanical properties in this project, where we basically could confirm different biomechanical properties of those cells as well. And another way of measuring this is by a colony assay in a 3D matrix. There was embedded in this matrix where we actually saw that they have larger colonies and not more, but therefore larger colonies which are eventually fused together. So we concluded from that keratins are crucial to keep this classical epithelia phenotype. Otherwise, keratinocytes are prone for EMT process. Now, this is a kind of the final slide of my PhD project. After this, I kind of switched a little bit the topic. I still stayed in skin research, but I got more interested in the environmental effects of the skin. Basically, as I said in my introduction, the skin is protecting us from a lot of environmental factors. And one of those factors, which is not very well investigated, is actually humidity. And my research goal of my project is to understand how stresses, in particular environmental stresses, are actually working on keratinocytes on a molecular mechanism way. Because what you do probably know is, if you're just listening to the cosmetic industry apps, you hear all the time that moisturizing is good for the skin, that moisturizing is helping you. And now we are approaching winter, so we have not only a more drier environment outside, we are actually also facing a lot of dryness inside of the room. So this is causing quite a tremendous stress for keratinocytes. And if you don't have any pronounced skin diseases, your skin is most likely reacting to this. It has been shown that the skin barrier can be increasing if you stay longer in drier environments. However, there are a lot of patients which are actually suffering from this dryness. They will tell you that their symptoms get worsened. And for some skin diseases, dryness can be actually also a trigger of the outbreak. So, but what is actually happening on the molecular level? So if a normal keratinocyte is exposed to dryness, the first thing what happens is that the cell loses water. And then the cell is shrinking together, and this increases the iron content of the cell. But it is inducing basically several mechanisms. So first of all, it's inducing hyposmolarity stress. by the water loss and the higher iron concentration, but it is also inducing a mechanical change of the cell. This is very important to mention. And this hyperosmolarity state we can actually also introduce in vitro by using several solvents which can mimic this hyperosmolarity stress. And then what are other reasons when the skin is exposed to this stress? There are transient reasons. So, for example, a shorter or longer experience of a dryness itself can be such a reason, but also a disruption of the epidermal barrier. A very simple example is wounding. If you cut yourself, then one of the first things what is happening is because of the cutting, you defect your epidermal barrier, and then you also lose water. You also cause a mechanical damage. You eventually inflame even your skin. That depends a bit on the circumstances, but overall, the water loss will occurring. And then there is a large list of reasons why you can have a chronic defect in the epidermal barrier. In EB patients, that would be, by the way, also the case because of the constant wounding of this blistering. There are other diseases which have a very high prevalence, which is atopic dermatitis and psoriasis. And both of these diseases, the skin barrier is already defected before anything happens. So they continuously lose more water than in an intact skin would happening. And then there are also these large reasons of chronic wounding. If a wound cannot be closed for whatever reason, you also continuously lose water in those cases. Just a little summary to atopic dermatitis. This is a skin disease which has a relatively high prevalence, up to 20% in children, where we have a chronic relapsing form of skin inflammation. The reason of the skin disease are not well understood. There is most likely only a minor genetic component to it. There are a lot of causes from the outside. Most of them are allergic reasons. So we also see in the patients EGA-mediated sensation to food or environmental allergens. And one thing which is crucial for the patients is that, or what we see in all patients, is that the epidermal function is defective and that we see overall a more dry skin. And what this is doing, or what does it mean for the patients, you can basically see here, you have this very dry regions and this dryness. And in general, this disruption of the skin barrier causes bacteria, viruses, and also fungi to get into the skin, and this causes an inflammation, and especially it causes an itch, which is quite a big problem for children because they usually scratch themselves and then they generate this wounding. And you in histology, you have as a hallmark this hyperproliferative skin, which is caused by this infiltration of the immune cells, which activates keratinocytes continuously to proliferate, and then they cause this really thick epidermis, which should help the patient to be more protective, which is not really the case for the patients itself. But what, besides this moisturing, which is helpful for sure for the patient, is actually underneath? What is actually the molecular mechanism? And to my surprise, this was not very well studied at all. So what did we do was we had a control wild-type model and receptor 1 and 2 FGF receptor model. And this, we were taking a low and high immunity. And then we did a proteomic analysis and wanted to figure out what could be potential changes in the proteome. And we found a protein which is called CSCA2. This was upregulated in those mice. And why did we took this mice? Was simply because this mice reassemble atopic dermatitis. They have a skin barrier defect. they show differences in the T-ring junctions, which are important for the cell-cell contact. They have continuously inflammation, and they especially have dry and fragile skin. And so in those mice, we could see that CSCE2 is upregulated compared to the wild type, and then downregulated if you put exactly the same mice in higher immunity. And it was really interesting to see, because this classical hyperproliferation, this classical hypersickening of the epidermis was just rescued just by the fact that they were exposed to higher humidity. Yeah, exactly. And now in my next couple of slides, I will just summarize a couple of results we've done over the years. So I'm happy to answer detailed questions in the end of my talk if you have any. The first finding we had, the first major finding we had, was based on this in vivo results. We were studying this now more on the human side of you. So we started to focus on human keratinocytes, where we saw that if we expose keratinocytes to hyperosmolarity stress or to low humidity, we also using 3D models of the skin, we see that this is induces CSCE2. And why do you need that? So the first thing which we done was just deleting CSCE2 or reducing the expression of CSCE2 in those cells. And we're looking at what do the cells do. And what we found was under non-stress conditions, CSCE2 reduced expression was fine. But as soon as we stress them with hyperosmolarity, then the cells have a survival problem, and they actually induce apoptosis, and they eventually die after longer exposure of hyperosmolarity. And why do we think they do this? Is CSCE2 was crucial to stabilize ecadherin at the membrane. And if you stabilize ecadherin on the membrane, ecadherin is part of the T-ring junctions, therefore at the cell-cell contact, you make sure that they can keep the barrier and they can keep the neighboring contact between the keratinocytes, which is crucial also for the survival of keratinocytes. Now, what do you need to know about CSCE2? CSCE2, as I started, was actually not a very well studied protein. It's probably also not very well studied still today, but we kind of helped to get more insights into keratinocyte functions. So CSCE2 is interestingly a transmembrane protein, meaning it is sitting on the membrane of a cell. It is cleaved. It is only expressed in epithelial tissues, and it was a known tumor breast cancer suppressor protein for breast cancer, sorry. And yeah, so this is all we wanted to start off with. And then over the years, we got a better idea what the function of CSCE2 in keratinocytes is. As I told you, we could show that it stabilizes the T-ring junctions. We also now know that it's actually also important in signaling. Ecadherin, part of the T-ring junctions, is interacting directly with beta-catenin. We later on could also show that CSCE2 is directly interacting with beta-catenin, and therefore it can change the Wnt gene regulation. One of the major functions of CSCE2 is really the stress keeper function. So as soon as you are under hyperosmotic stress, it will promote survival under this conditions. And CSCE2 overall reduces migration. This is also a very important function of CSCE2. And then we went further on and looked into more other aspects of gene regulation and signaling, and also of biomechanical properties, because just as a short reminder, the cytoskeleton I introduced in the beginning is actually connecting cell matrix adhesion to cell-cell contacts directly. And so it was just a matter of question, could it also be that it's not only cell-cell contact which is altered, could there also be a difference in cell matrix contacts underneath? And one result which was really surprising and kind of changed the project in a very different direction was, even though that CSCE2 is a plasma membrane protein, I made this finding that we actually found it in the nucleus of the cell, which was, like I said, rather surprising. we, from there on, started to study the nuclear function of this protein. Further, this LMB, this is it stands for leptomycin B. This is an inhibitor of nuclear export, and nothing can get out of the nucleus anymore. Everything gets enriched, and if we treat the cells with this inhibitor, then we see a sudden enrichment of CaCe2. But we also used an experimental trick where we deleted the NLS. The NLS stands for nuclear localization sequence. And if a protein doesn't contain NLS anymore, then it cannot be directed to the nucleus any longer. Of course, we proved this. We did several experiments to confirm this and to confirm that it cannot be trafficked to the nucleus and so on and so forth. So part of it is shown here, but there are also additional experiments to prove that. And then we wondered if CaCe2 cannot go into the nucleus anymore, does this actually alter the function of CaCe2? And that is indeed the case. So we can actually show that if we re-express either the wild type or the NLS mutant in CaCe2 knockdown cells, then this changes completely the function. So we can rescue the survival function with the rescue of the wild type expression of CaCe2, but we cannot do this if we just express the NLS mutant of the cell. And also in migration, we see differences. I told you if we have increased levels of CaCe2, this reduces the migration. Vice versa, if we have less CaCe2, we have an increase in migration. So they move faster. If we now not take the wild type CaCe2, but we take the CaCe2 which has this mutation in the NLS, we see that the migration cannot be rescued completely and is significantly different compared to the wild type CaCe2. So it seems that the nuclear CaCe2 really has an important function of the cell. And there is this very big question, how can actually a transmembrane protein be transported into the nucleus? And a way of doing this is by secretion, or basically just keeping the protein on a membrane. And CaCe2 was hypothesized for quite a long time that it could be eventually secreted. And we then had a cooperation with the drug delivery LaRue lab at ETH, and so I started to learn more about extracellular vesicles, and we found that CaCe2 is part of the extracellular vesicles, but we can also show that extracellular vesicles are a way of trafficking the cells inside of a cell, but more importantly, also outside of a cell. So extracellular vesicles are secreted by all cell types in the body. What one does know is that we express more extracellular vesicles under stress and under disease state. But I have to add that this is a relatively young field, so there are a lot of open questions about this. One idea is to take extracellular vesicles as potential biomarkers, because extracellular vesicles are quite stable because they have a double lipid structure, so they can actually also go to this bloodstream and be trafficked all over the place. And but they also can locally tell neighboring cells there's something going on. It's believed that extracellular vesicles are kind of signals saying, okay, I could need help here, I give you some extra signals, but this is still a bit unclear what exactly the function of extracellular vesicles is. We do know that it's more promoted in wound healing, it has a function in hair growth, it is different in skin diseases and skin pigmentation, and so on. And there are even some first clinical trials done with extracellular vesicles for chronic ulcers, burn wounds, and also dystrophic epidermolysis bullosa. But I have to mention that there are clinical phase 1 studies, so very, very, very early on in the clinical phases, but it's a quite promising field. And that's how extracellular vesicles are looking like. Again, you can see here this double lipid structures, which we are isolated. And something which I didn't mention before, but extracellular vesicles can contain everything. So basically DNA, RNA, proteins, transmembrane proteins, channels. There are really almost daily new studies coming out which have been doing proteomics, transcriptomics on extracellular vesicles. And we are kind of studying extracellular vesicles under different conditions. In all, also not only in skin, but also on other aspects. This one is quite a full slide, but I will make it as simple as I can. So what is important here for those studies we did is that CEC2, we could show here that it's secreted in extracellular vesicles. And these extracellular vesicles are very useful because they can be uptaken by the cells. And with this process, we believe this is one way how CEC2 can enter the nucleus. There are eventually others, but we could show that extracellular vesicles are one way of proteins to go there. And one thing I would like to add here is this mechanism is probably not specific for CEC2. There could be also other plasma membrane proteins which could use this trafficking process and therefore can cause changes in the gene regulation or in general in nuclear processes. For CEC2, we could show that nuclear CEC2 is important in Wnt target genes for increased survival under hyperosmolarity stress and also for reduced migration. And then we changed the experimental model a bit. We was going from a knockdown of CEC2, which left the residual expression of CEC2, meaning there's still a little bit left of CEC2. And we know that this little bit can actually help the cells quite a bit. And therefore, we decided to delete CEC2 completely for keratinocytes. And the method of choice, how we did that, was a CRISPR knockout, CRISPR-Cas knockout. And here we, first of all, recapitulate all the results we know from our experiment models before. So we can show that if we don't have CEC2, we get higher apoptosis rate after hyperosmolarity stress. We see that knockout cells of CEC2 are much faster in migration. But we also saw that the cell matrix adhesion is severely altered. And that effect we haven't seen as much with knockdown cells before. So that was quite a new phenomenon we saw. And we studied this with the help of the Zambelli lab at the ETH for biomechanics, which is a biomechanical lab, which uses a so-called fluidic force microscopy. And this is actually quite a fancy method to measure adhesion forces, so how tightly a cell is attached to a surface. And what we have is you have a cantilever, which is full of liquid, and that kind of sucks on the cell. And it sucks as long as it can detach the cell from the surface. And this force which is applied is relatively mild and gradual, and it really looks for the force which is needed to detach the cell. And this is how it looks like. So it attaches and then it sucks, basically. And if you remember the way the cell attaches is on two ways. On one hand is focal adhesions, on the other hand is hemidesmosomes. And you can think of that there are first a few focal adhesions detached, and then the entire cell is attached as a complete cell. And that's actually also something we can see in those curves. I will not go into detail with that, but you can see that it's first minimal forces are needed, and then you generate the entire complete detachment of the cell. And how does it look like experimentally? So you have here this cantilever approaching this cell here, sucks it relatively slowly in. And then here in those images, you can see that the cell is actually completely sucked in this cantilever. And then you measure, by doing this, you measure the force, the energy, and several other factors. And what we learned is using a system where we brought back CEC2 inducibly. That means we have our knockout cells, no CEC2 in those keratinocytes, and then we use stably but transiently we can switch on CEC2 expression, if you like, with doxycycline. Without doxycycline, we don't have any CEC2 expression. If we add to those cells doxycycline, CEC2, so the promoter is switched on and thereby CEC2 expression is induced. And if you do this, then we see that the cells, you need more force to detach the cells from the surface, basically. So meaning CEC2 makes this adhesion force stronger, and it's actually needed for adhesion. And if you continue here, what we also know is that if we lose CEC2, we actually destabilize focal adhesions quite a bit. So you can see here focal adhesions have a completely different structure than hemidesmosomes. They are this tiny thin line structures, this patches in the end of a cellular membrane, of a lamellipodia of a cell, as you can see here in this wild type, so with all these arrows, which are shown here. And then here in the knockout CEC2 cells, you first of all have even problems to find focal adhesions, but if you do, then they are quite tiny and not so mature as they are in the wild type. We can also quantify this. We can quantify this depending if CEC2 is turned on or not. And we see in this experiment quite some similarity to the force we need in the fluidic force microscopy. This is a very interesting finding. And really, to very briefly, and I'll make it as easy as possible to summarize these findings we did in this study, we can see that CEC2 is interacting with beta-4 integrin, which in turn interacts with other integrins like beta-1 integrin, which is so beta-4 integrin is a major integrin of hemidesmosomes. Beta-1 integrin is a major integrin of focal adhesions. And it is known that they interact together, and it's known that integrin induce a signaling process. So it's just not something happens at the cell membrane. No, there's actually tight regulation of attaching to the extracellular matrix. And if this is changing, this is also changes the signaling internally of a cell. This process is called mechanosignaling. And not going into further details than that, but we can show that CEC2 can alter this focal adhesion mechanosignaling on the cells. And we can also show that if we add to those cells extracellular vesicles, we can actually rescue this effect. And so extracellular vesicles seem to be very helpful for adhesion and migration processes here. And in summary, what I tried to convince you about today is that CEC2 is a very important protein, stress protective protein in the skin. It is important for survival under stress. It's important for migration. It can alter the signaling. It can alter cell and cell matrix adhesion, and also changes biomechanical properties. And one big outlook we are now looking into is what do extracellular vesicles actually do in the communication in skin. And here we don't only focus on the epidermis, but we also focus on the dermis containing fibroblasts. And we really want to understand how they promote this crosstalk between each other. And we want to learn more about the uptake of the extracellular vesicles and also of potential changes in gene regulation. And with that, I would like to acknowledge, first of all, my time in Thomas Magin's lab at the University of Leipzig, which was a very great experience for my PhD. And Professor Svane's lab, where I continued my postdoc, and now my time as a senior scientist, and all the people. There have been over the years a lot of people involved with this. We have several collaborators here, but also outside of ETH. Last but not least, I also would like to mention my founding. And yeah, thank you for your attention, and I'm happy to answer questions.
Thank you so much. That's absolutely amazing body of work. I don't even know where to begin. I started making some notes about how interested I was in keratins, and then I've made some notes about how interested I am in extracellular vesicles. So, well, if people want to put a few comments in the chat, or if you want to use the Q&A, please go ahead. I don't know if you can see the Q&A, Kristin. If you
Not
can't,
yet.
I
I
can.
need
Yeah,
to get out of the talk, I think.
sure. But if people have any comments, do go ahead, because otherwise I will just ask a few. Um, I might have missed it. I know you were doing the knockout cells for the CLCA2.
Mhm.
Is there a mouse, a knockout mouse?
No, there's not, because
Mm.
the reason why we changed to the human system is that maybe this is one of the few cases where a protein is more complex in mouse than in humans. So we have more CEC2 variants in mouse than in humans. And yeah, it was just,
Okay.
it is anyway
Complex.
important to look into human function.
Yeah,
So, but it's also easier to
yeah.
do this in humans, but we
But
can not generate a knockout human, of course. That's
you said it's a transmembrane protein that also appears in the nucleus.
yeah.
Is that common? I'm thinking back here. I used to think of them as separate things. You have nuclear factors, you have transmembrane factors, and it's not. Is it not? It's not that common for proteins to do both roles, is it? Is that an unexpected or
It is
unusual
kind of
finding?
unexpected, which I think is mostly coming from the fact that the fields are working a bit um, yeah, you specialize in a certain kind of field, either on the plasma membrane or on the nucleus, but you kind of stick in your compartment. And at the time when we first started to see that CSC2 is going to the nucleus, I went to several nuclear conferences to kind of learn something about it, because I also was new in that field. And there was one talk I remember very well where somebody showed a nuclear proteome of a cell, and they actually showed that 30% of these nuclear proteins they find are plasma membrane proteins. So it seems not to be uncommon. What is uncommon is studies about them. I think this is still a field
Okay.
which is a bit understudied. Yeah.
I think you're right. People stay in their own compartment and then it's that
Yeah.
overlap.
Yeah. Yeah.
And I guess, so are the extracellular vesicles part of this? Are they transporting, mm, so the CLCA2, is that in the extracellular
Yeah.
vesicles?
Yes, it
Yeah.
is.
And presumably that's helping it to travel to other cells, but
Yeah.
within a cell, is it staying in that same vesicle to be transported to the nucleus?
Yeah, that's what we think. So what is
Yes.
happening as an uptake process is we think it fuses to the endosomal compartments, and then it traffics with the help of the endosomes to the nucleus.
Yeah.
For the endosomes, it's also a bit more studied than for extracellular vesicles. So it's more studied inside than to
And
have this
outside.
idea that the vesicles could also eventually come from the outside, actually.
It's really interesting. So what you've described with the extracellular vesicles, presumably the contents will differ depending on the type of stress a cell is
Yeah,
under.
yeah, yeah, yeah.
Because if you're doing the clinical trials are in progress,
Yeah.
well, they seem like a sort of like people use lipid nanoparticles for gene therapy. Are they the body's own way of doing that, moving things around between
It's
cells?
basically, so extracellular vesicles are a bit more approached lipid vesicles, and they are also, we are having this cooperation with the drug delivery lab, and they actually can load extracellular vesicles with drugs. And their research is about that. Their research is about how can we use extracellular vesicles for drug delivery system, which is a bit easier in skin because we can apply this topically. But for other organs, I would like to say that we are not so, we don't know yet how we can actually promote the extracellular vesicle to go to the place where we want to go it
No,
to.
it's skin we're interested in. So if it's
Yeah,
a
yeah,
good
yeah.
system for
So
skin,
that's easier,
yeah,
right? So if you have a wound, you can apply them topically and
yes. Oh, that's very interesting.
and
Yeah,
help. So yeah. But
yeah,
we
that's
know that
brilliant.
there are so many extracellular vesicles secreted in wounding, for example. In this
Well,
wound
that's what I was.
fluid,
Yeah,
you
yeah.
can measure them a lot. Yeah.
Yes, just very interesting for people living with EB that could be a process that could be taken
Mm-hmm.
advantage
Yeah.
of. Yes,
Yeah. Yeah.
and there aren't hordes of questions coming in, so I'll just ask a couple more maybe, and if there's any final thoughts, please get your questions in now if you want to. Just going back to the keratins, which, and this is maybe a stupid question, about, it's the end of the webinar, and I'll get the comms team to cut it off if it's a very stupid one. But about keratins and the different types, and I'm asking about a degree of redundancy. How easy is it for one keratin to sort of swap in with another one? And then my really stupid question is not really a scientific one, but I'm hearing so much from people who are taking keratin or collagen supplements. All of my biology training makes me think that there's no way you could eat a keratin and then have it turn up in your cells. But it seems to be what a lot of people are believing. So yes, if you could, I'll just ask you about the redundancy really of keratins within cells, but also if you've got any comments on keratin supplements, I'd be
So the
interested.
second one is more tricky because I may miss data here. And then the thing is, the second one is actually, in general, the problem is that we miss a bit data. For the first one, it's also not so easy to answer. We do know that a keratin expression changes if, for example, cells or the skin is, so let's take this nice example of wounding. If skin is wounded, then the keratin expression changes to K6 and K16. They are so-called the stress keratins. So how easy this is that they alter keratin filaments, we actually, I would say, I think Thomas would be the much better person to ask this, but I would say it's not so easy to answer, but it is possible. It is probably rather, okay, my favorite partner is not here, I have to take another one. But we know from AB that it's not so easy, right? So if there is a mutation, they just fail to assemble these filaments, which they actually need. And for the second one, I, for a lot of reasons, don't want to speculate too much, but let's say I don't take any of this.
Likewise, I guess it's a question for a nutritionist, maybe, or a dietitian.
I have my doubts that we can take, so especially keratins are proteins which are hard to dissolve. So it's really difficult to get them in solution. With everybody who ever worked with keratins knows that actually. So I don't see any benefit from.
I have to be, I'm in agreement.
The same is true for collagen, by the way, but collagen are very large fibrils, which are also very difficult to get in this structure. Because one part I would like to add here is, I think, what is a bit forgotten by the general public. One thing is if you have a small peptide, and the other thing, if you have the final structure, and this matters a lot for keratins, and that matters a lot for collagens as well.
I think, yeah, that's good to hear. I think a lot of the supplements talk about being hydrolyzed anyway, so they're already broken up into small pieces to get.
Yeah, they are. Yeah, they have to, because otherwise they cannot get in to us. So.
Thank you. I'll pass that on to my sister, but thank you. Listen, there haven't, as far as I can see, I don't think there are any other questions that have come in. It's been a really fascinating talk, and thank you so much for taking the time to share all of this work with us and for the work that you're continuing to do. Oh, hang on, we got one. We've got one more here. I don't know if you can see in the Q&A, we've got a question. Do you think nuclear CLCA2 could control keratin expression or regulate keratin assembly?
We haven't tested that at all. So what I have done is I have looked in the keratin knockout cells of the filaments, so actin, tubulin, and keratin is differently structurally. And I can say actin is. For keratins and tubulin, we don't have any differences seen, but I have not studied it in really, really great detail. And
there's more of the story, more of the story to come together, I think.
Yeah. That makes sense, but we actually have not concentrated on this yet.
Brilliant. I hope you get the chance to do that and can report back to us maybe in a few years. Absolutely brilliant talk. Thank you so much for taking the time. Thank you to everybody who attended. You will get an email probably asking you for some feedback. But brilliant. Thanks very much, and enjoy the rest of your day, everybody. Thank you again. Thank you again, Dr. Seltmann.
Have a great year.
Thank you. Bye-bye.
Bye-bye.